Hello students, as you're watching this video, you're going to get a glimpse on what is DNA barcoding. This world is our home. We live here and share a home with millions of other species, most of which we know nothing about. All these species of plants, animals and insects we see around us every day are closely connected to each other. Yes, we are influenced by and influence a number of species in and around us. As in a society, we cannot live happily without knowing each other. It is equally important to know and identify every living neighbor or species that share a home on planet Earth. It becomes impossible to name and classify the approximately estimated 8.7 million plant and animal species which live in different forms and stages on our planet Earth using traditional methods. Using advanced DNA-based tools like DNA barcoding, it becomes possible to identify and classify the innumerable number of known and unknown species in and around us. A barcode you see on your identity cards, grocery items, and library books every day carries a binary code. If 1 and 0 can form innumerable barcodes for infinite items in the world, scientists thought, why not a barcode with A, G, T, and C, namely adenine, guanine, thymine, and cytosine, which are the 4 primary letters that form the genome, which is predominantly the DNA content inside the cells of every living creature. Yes, DNA barcoding can be defined as a standardized approach to identifying plants and animals by a short DNA sequence from uniform locality on a genome. The concept called DNA barcoding was put forth by Paul Herbert, who proposed that short DNA sequences can be used as barcodes. There are 5 basic steps in DNA barcoding. Number 1. Specimen collection with a voucher number. Number 2. Genomic DNA extraction. Number 3. PCR amplification of specific targeted DNA regions of the genome. Number 4. Sequencing of the barcode. And number 5. Publishing it in any one of the DNA databases like NCBI and get a GenBank accession number. Usually, for animals and insects, the standardized DNA barcoding region is the COX-1 or cytochrome oxidase gene. For fungi, ITS or internal transcribe spaces are predominantly targeted. And for plants, a number of cpDNA or chloroplast DNA regions, either genic or intergenic spacer regions, are targeted. This barcode is not the conventional binary barcode, but a DNA barcode. Yes, a DNA barcode for every living creature that sojourns around us every day. So dear student friends, whenever you see an organism that has life in it, remember that it carries an identity as a DNA barcode within its cell itself. Hope it was interesting, and thank you for sparing your valuable time.